In this section, I want to talk a bit about how the workflow of non-target analysis actually looks like. What is illustrated here in this slide is a workflow that basically how you can view how non-target analysis generally is considered. So you have initially the sampling, which is actually a very important aspect. So when you think about where do you sample, how often do you sample, then as a next step, you have the actual analysis. And for that, you have to think about, do you, for example, need to extract the sample? What type of analysis do you need to do? So, for example, do you need to do gas chromatography, high-resolution mass spectrometry, or liquid chromatography, high-resolution mass spectrometry? And this is followed then by a very extensive data processing, which is very important, especially for non-target analysis, to actually detect the different peaks of the compounds that are present in your sample. One thing that is also very important is to think about what is called blank or background subtraction. So here, what you account for is, are there any background contamination that originate, for example, just from the lab that you're working in? And if you want to assess exposures, you want to make sure that you can subtract these, because you're not interested in kind of like what's actually in the lab that you're working in, but what are the compounds that are really originating from your sample. Another aspect that is often important is the use of replicates. So not just use or analyzing a sample a single time, but also typically several times. And what this allows you also is to gain more confidence in, like, are your compounds really present? Or are there any issues, for example, with the analytical tool? And then this is typically followed by a prioritization approach. And this also really depends on the study or the research question that you're asking. So this can, for example, be to investigate certain spatial or temporal trends. So when you think, for example, about chemical exposures, where you know chemical exposure is only occurring during a certain time window, you could take a sample before that exposure occurs, during the exposure, and after the exposure, and you can basically look in your data for these temporal trends. So that's one example of a prioritization strategy. What you might also do if you're looking for specific chemicals, I had given earlier this example of the use of bleach and the formation of chlorinated compounds. You can also look for specific isotopes. So chlorine, as an example, has two important isotopes, chlorine 35 and chlorine 37, that can both help identify certain compounds because you can look for these isotopes. But again, they can also help to focus on, specifically in this case, for example, on the formation of chlorinated chemicals. And then you can also use advanced statistical approaches such as principal component analysis, or PCA, or clustering analysis, that can help you identify different clusters or associations between different samples. So for example, if you have two different cohorts that you're looking at, so for example, let's say hairdressers and office workers, you can use these statistical approaches to see if you can also see this difference in the results of the analysis. And then last but not least, the end goal of most non-targeted analysis is really the identification of compounds. And for this, we typically rely on the use of online databases. We also use suspect search. As I mentioned earlier, suspect screening is typically used if we look for compounds that we suspect to be present. So for example, I had given the example of using hair products or ingredients of hair products. Then we can also rely on the information that we're getting from the high-resolution mass spectrometer, so the mass of the ions that we detect, which is indicated as MS here, or also the fragments, which is indicated here as MS. And then last but not least, if we really want to ultimately identify a compound, what we typically do is we use reference standards. So in most cases, we buy a reference material. You can also synthesize them in the lab, even though this is obviously very time consuming. And if you use these reference standards, you can, on the one hand, really confirm the chemical structure of a compound. So you can really, with 100% certainty, say, okay, this is the compound that I think it is. But another important aspect is also that you can actually quantify. So you can determine the actual concentration of a chemical. And this is really important if we think about risk assessment and the concentrations and the potential risks that arise from these exposures. Due to the large amounts of data that occurs in the large amount of compounds, one very critical aspect that is very important to consider, and I would say that takes a majority of the time also in the data analysis, is quality control. Due to the sensitivity of these high-resolution mass spectrometers, we detect large numbers of compounds, and we really want to make sure, if we are thinking about exposures, for example, that we can, with a certainty as high as possible, say, okay, are these compounds really coming from the exposure, or is there a potential issue with background contamination? So for example, from the laboratory where the sample preparation is performed, potential impurities in any standards or solvents that are being used during this whole process. And then another aspect that is also important to consider is what chemical leaching that can come from certain equipment, but also, for example, the plastic bottles that are used. Another important aspect is also computational noise, which basically means these are artifacts that are coming from the data processing. So there's a lot of computational power that is behind the detection of these compounds using high resolution mass spectrometry. And it can happen that there are certain artifacts that are just caused by this process. So it is important to also consider for this. And then we also have to think about instrument variability. So that means variability that can happen from sample to sample, even within the same, if we run samples one after each other, or if we also run samples on different dates. There might be changes in the instrument, the sensitivity might be changing. And so we have to take these variabilities into account, because otherwise we might see some differences that are just caused, again, by these variabilities and not really a representation of differences in these samples. And so to do this, we use blanks, which basically are control samples that are just used in the lab. We use quality control or QC samples, which typically contain a mixture of chemicals that we know about. We use replicates, meaning, for example, if you need to take a sample at a certain location, we don't take just one sample, but we take several samples. And similarly, what we often also do is when we analyze these samples, we don't just analyze them on our high resolution mass spectrometer just once, but a subset of samples is typically injected or analyzed several times. And then another aspect that can also help to avoid any issues that are caused by instrument variability is just to randomize your samples, to also take that aspect out. And so when we think about the analysis, what I'm showing you here is kind of the result of how a typical output of a high resolution mass spectrometer, in this case, this is a chromatogram from a liquid chromatography high resolution mass spectrometer showing. So when I say chromatogram, what you see here is you have the retention time. So this is basically the time it takes for the compound to come out of the chromatography system, which is shown here on the x-axis. And then you have the relative abundance. So basically, you can think about it as the number of molecules that are coming out on the y-axis. So the chromatogram is a plot of the detector response as a function of time. And then what is shown here is a so-called total ion chromatogram that shows the total signal of all ions, or M over Z's, which stands for mass over charge, that we're detecting during the analysis. And then what we can do is we can basically look into this chromatogram at any given time and look at the mass spectrum, which is shown here, which is a plot of the mass to charge ratios measured at a particular time of a chromatogram. And so in this case, what you see here on the x-axis is actually the mass over charge ratio, so the mass of different ions at a given time, and then you have the relative abundance on the y-axis. And so here you can see that this mass 564.3583, for example, is very abundant. But you can also see that there's a bunch of other different ions present. And as I mentioned earlier, one of the great advantages of non-target analysis is really that we can detect large numbers of different compounds. But I hope it also becomes clear here how important it is to really take also into account, are all these ions really coming from the sample, or are any of these ions potentially coming from any background, either from the analytical instrument or from the laboratory that we're in. Once we've analyzed all of these samples, the next step then is really the data processing and really thinking about, okay, what are actually the compounds that are present in the sample? And so what we typically look at for this is the so-called extracted ion chromatogram, or XIZ, that shows now the signal for a specific ion or M over Z in the sample. And this is typically more useful than the total ion chromatogram that basically shows all ions that were detected. And the peaks, which is shown here for a specific compound, which you can see here a nice chromatographic peak at this retention time of 14.29 minutes. What the software now does, it can basically integrate the peak area that is under this peak. And this is important because this peak area is proportional to the amount of the analyte that's present in the sample. And so if we would have a reference standard of the specific compound, we could basically determine exactly what concentration this peak area corresponds to. One aspect that I want to highlight, and again, this is why looking at the data and really thinking about the quality of the data really matters, is that the software or the instrument doesn't necessarily differentiate between how well each of the ions was detected. So what I'm showing you here is 4 examples of what the instrument or the software detects as different peaks. So on the top left, you don't really see anything, which is kind of like the point. You see this kind of like very zigzaggy line that is kind of like just noise. And then when you go to the right, you see there is somewhat like what we would call a chromatographic peak. At the bottom left, you see this peak is even a bit more clear. And then when you go to the right, you can see this is really what a clear peak. And actually, the software allows us, and this is a very helpful tool, to actually determine the quality of this peak. And this is extremely helpful because you would probably agree with me that this top left here, assigning this to a peak or a specific mass would probably be very misleading. So having this peak rating, in this case, that the software actually is helpful to really think about, okay, which are the compounds that we have confidence in that these are really actual peaks. And so what you might say, what you can tell the software is, for example, to only consider compounds that have a peak rating of, for example, above 6 or even above 8. So where you really only focus on these compounds that are clear peaks. And then the next step, and this is really depending on the question of the study that you're interested in, is the prioritization of different compounds. And just to illustrate one aspect that can be very helpful. So for example, when you think about chlorination or chlorinated byproducts that are forming when you use, for example, bleach, is this chlorine isotope pattern. So what I'm showing you here on top is the detection of a compound. In this case, it's imidacloprid. Based on the sum formula, you can see that it has actually chlorine that is part of the molecule. And what is important when we think about chlorine, this is very helpful when we do this type of analysis, that chlorine actually has 2 different isotopes. One is chlorine 35 and the other one is chlorine 37. And what is important here is that we know that in chlorinated compounds that chlorine 35 typically is 3 times more abundant than chlorine 37. So when we look at the masses at this specific peak, you can actually see this. So you can see here at this mass 256.0592, which is shown here in the lower graph, you see this is one peak where you have the chlorine 35. And then here on the right side on the lower graph, you can actually see a second peak at 258 that is actually corresponding to the same compound. But in this case, the compound has the chlorine 37 isotope. And this is very useful because this ratio of these 2 peaks is actually a constant, and the software actually is able to tease out basically all the compounds that have, for example, chlorine in them, which is an extremely helpful aspect when we look into chlorination byproducts. So byproducts that are forming when we use bleach, for example, in the disinfection of surfaces, or it is also very commonly used in drinking water treatment. One aspect that I wanted to briefly also mention is that there is different confidence levels of the compounds that we detect. So not all compounds that are detected, we know at the end with certainty that they are the compounds that we really think they are. And to differentiate between these different confidence levels, Emma Szymanski and colleagues published a paper in 2014 in Environmental Science and Technology where they proposed different confidence levels. And so the lowest confidence level is the level 5. So in this case, we have a mass that we can detect, but we don't have any matches with any libraries, online databases, or libraries that we might have in-house. So this can either mean that the software, so in this case, we're using Compound Discoverer, abbreviated as CT, doesn't give us a prediction of a formula that's present in online databases. Or what can also happen is that we get a prediction of a number of different formulas. So we don't really know which one of those it actually is. When we go one level up, level 4, in this case also we don't have any spectral library matches, but we have a single predicted formula that we're getting from Compound Discoverer that is also found in, for example, online databases such as ChemSpider. Level 3, which is the next level, means that we actually have spectral library matches. So the compound is included in spectral libraries, but we don't have just one match, but we have several matches. And you can distinguish those with the quality. So basically, when we think about spectral libraries, we're thinking about different fragments and the number of different fragments that are kind of matching with the compounds that are in the reference library. And so this is evaluated based on a score. The higher the score, which ranges from 0 to 100, the better is basically the match. As I said, level 3 is that we have multiple plausible matches, which is in contrast to level 2, where we actually have just one single match with the library with a very high score. In this case, we used a score of over 70. And this can only be topped basically by level 1, which is kind of the ultimate way of really unambiguously identifying compounds using a reference standard that you then match by analyzing it and determining that it has the same retention time or RT and also exactly the same fragments that you see in your sample. So this is the ultimate confirmation of your compound identity. And just to highlight again why this identification and to really unambiguously identify the structure of a chemical is so important is illustrated here in this slide. So what I'm showing you here is on the left side, you see these two compounds, 2-chlorophenol and 3-chlorophenol. And if you look at the chemical structure, you can see that they look very similar. The only difference is that the position of this chlorine is different. Now, if you really want to determine the difference, what you basically need to do is you need reference standards. So you need these level 1 confirmation. And you can probably imagine that it is very important if you, for example, think about the toxicity of these compounds, because the toxicity is likely to be different between 2-chloro and 3-chlorophenol. If you don't have reference standards, the way we typically deal with this is that we are proposing a tentative structure, which is level 2, which is indicated here on the right side, where we basically indicate in this case that with this horizontal line here on the chlorine, that we know that there is a chlorine present. And as I mentioned earlier, we can do this, for example, by chlorine isotope pattern detection. But we don't really know exactly what the location of this chlorine is. So it could appear right next to the hydroxy group, or it could be in any other position of this ring system that you see here. Okay, and then last but not least, the ultimate goal is really the identification of compounds. For this, in addition to having reference standards, there's also an increasing number of computational tools that are important here. One important tool is the ChemSpace mapping tool that has become available recently and that is still being developed or further developed. This is a very important tool because it provides information about the physical chemical properties of a chemical. So what this means is it, for example, allows software to determine the polarity of the compound and other aspects. And this is very important because it can help to determine if a compound can actually be detected by a specific analytical approach. And it can also help to determine if a compound can actually be present in a specific sample matrix. So just to give an example, if you analyze an air sample and you just are interested in compounds that are present in the gas phase, you need basically chemicals that are volatile, so that can be volatilized and be present in the gas phase. So you can use this characteristic to narrow down chemicals that are actually volatile and that you might be able to detect in this specific sample matrix. Another important aspect is what is called in silico fragmentation. So this is basically using computer software to predict the fragmentation of a compound. In addition to the detection of the masses of ions using high resolution mass spectrometry, the fragments provide important information about the chemical structure. And if you don't have a reference compound, either a reference standard in the lab or a data point in a spectral library, you can use these in silico fragmentation tools to get a better prediction of if you fragment this compound, is it really leading to the fragmentation pattern that you're seeing in your analysis? So this adds a level of confidence into the identity of the compounds. And then one other aspect is also when we think about the exposure to chemicals that can also really help is the prediction of metabolites. So if we're exposed to a certain chemical, for example, through air or water or skin, we can use these software tools to predict the potential metabolites and then match these metabolites with what we're actually seeing in our sample. And this is important because sometimes if we are exposed to certain chemicals and these are extensively metabolized in the human body, we might not actually see the original compounds, but we might only be able to detect these metabolites. So if you want to assess exposures, we need to monitor also for these metabolites. And so if we want to predict these, there's a general approach that you could use where you use, for example, cell or microsome incubation. So these are parts of cells that are responsible for the metabolism of chemicals, where you incubate basically your chemical of interest and then look for metabolites. This is a very time-consuming and expensive process. So what you can also do is actually using prediction software tools, such as those that are listed here. Biotransformer and SciProduct are two of them that allow you to predict potential metabolites, and the information on these predicted metabolites can then be directly used in your non-target analysis workflow. So for example, to include in the suspect screening list to specifically also look for these metabolites.